Yes, welcome back to my channel. Today is the third session of the zoology. Before we had done the endoplasmic reticulum and the plasma membrane. So today's session is Golgi apparatus or Golgi complex, also known as lipochondria or idiosome or Baker's body. It is a cell organelle. Basically, Golgi apparatus is a cell organelle and present behind or opposite in direction to the endoplasmic reticulum. Then the Golgi apparatus was invented by the first Camillo Golgi, hence the name is Golgi apparatus in 1898. Owl and cat nerve cells are found, in which the Golgi apparatus are first discovered is in owl and cat nerve cells. Observe the Golgi apparatus. What is the diameter? The diameter is about 0.5 to 1 mm. Micrometer. Size may varies. Present in nerve cells or neurons and liver cells, hepatocytes, and then the basic to endoplasmic reticulum. So the cisternae, as I told before in the before session, if you had watched the video, what is the cisternae? The folding like structures called cisternae. It is of about length 0.5 to 1 mm in length, and these are flat in Golgi complex. And these have a disc-like structures at its ends. It resumes like a disc-like structure and parallelly arranged and look like a bone. Then the transphase, protein synthesis and releasing, in which the protein gets synthesized and released. This phase is called transphase. What is the cis phase? Here are the two phases, cis phase and the transphase. So cis phase receives the vesicles. Vesicles are the rounded like structures present on the cisternae. The two vesicles are present. So it receives the vesicle, proteins and sugar are present. It receives vesicles in which in the cis phase the proteins, sugars are present in cisternae. Then the protein enters the cisternae. The entering part is called lumen. Protein plus sugar form glycoprotein. This is called the modification, in which the protein and sugar gets modified into glycoprotein. Then the protein plus lipid modified into glycolipids, from where the protein and lipid is coming. As we know before, the wall of the cell has two components, made up of two components, proteins and lipids. So then the maturing phase or the maturation phase, in which the vesicles are present or secretory protein. It's also known as secretory protein. If body requires protein, vesicle goes out from the Golgi complex carrying the modified proteins, and then the tubules. What are the tubules? As I told in the before session, for the detoxification, tubules are present on the endoplasmic reticulum. So cisternae are interconnected. Cisternae are not the separated, they are interconnected with each other. So the tubules are present for interconnection to connect between or to transfer or to transport between the one cisternae to the another cisternae. The protein, lipids, glycolipids, glycoproteins get transferred. For this process, tubules are present. and it forms the vesicles. The vesicles are small and smooth. Then what are vesicles? There are three types of vesicles. As I said before, the rounded structures which are present on the cisternae which carries the protein or the modified protein are called vesicles. So the first type of vesicle is transitional vesicle, or it is called smooth vesicle. Then second, secretory vesicle, or called rough vesicle, and the third, clethidium vesicle. So let us see one by one. Transitional vesicle. Smooth layer. The layer or the upper layer which is present is smooth in nature. The vesicle releasing from transphase called transitional vesicles. The vesicles which release or which moves out from the transphase of the Golgi complex and received by the Golgi complex of cis phase. So it goes out from the trans phase of the Golgi complex and receives by the cis phase of the Golgi complex. This is called transitional vesicles. Then what are the secretory vesicles? The outer layer is rough in nature, so we called it as the rough vesicles. Here the vesicles releases from the transphase of the Golgi complex or from the maturation phase. And then the clethidium vesicles. Vesicles transferring through or from the tubules called clethidium vesicles. As I said before, what are the tubules? These are the interconnection between one cisternae to another cisternae from which the protein or the molecules or ions get transferred from one cisternae to another. Then what are the vacuoles or the Golgi vacuoles? These are the single layered. These are the structures formed by the Golgi complex. Here the vacuoles and the lysosomes are present. As I said before, what are the lysosomes? Helpful for breakdown of food or which helps in the digestion of the food. These are the enzymes, and these are the hydrolytic enzymes, and it releases the hydrolytic enzymes. The lysosome is very important for the digestion of the body. Then the vacuoles releases enzymes and producing. Vacuoles produces the enzymes and releases the enzyme, hence the vacuoles are very important and it plays a very important role. What is the chemical composition? Proteins and lipids are formed by Golgi complexes. Proteins and lipids formed by Golgi complexes. Modified protein is required when cell damages and hence it repairs damage. What are the lipoproteins? Lipoproteins are those which are made up of enzymes like ATP, ADP and amino synthetase. Then coming to the functions. What are the functions of the Golgi complex? Here the main functions. There are a lot of functions, but I had take the important functions only. So let us discuss. The first function is the formation of vacuoles and lysosomes, which releases the enzymes, which plays a very important role in digestion of food. And then not present in sperm in early stage. The Golgi complex is present everywhere as we know, but it is not present in sperm in the mature phase. Sperm is developed after development. There is no Golgi complex in the sperm and ovary and also in the RBCs. But during early stages of the sperm development, Golgi complex is present, which form the acrosomes, which is helpful for penetration of the ovarian membrane. And then the third, in plants during cell division, formation of cell plates through which the translocation takes place, plays a major role in the formation of cell plates. Then the fourth, formation of cell wall in plants. And then the fifth, acts like a traffic police for the modification of proper cell. It releases the required amount of proteins, modified proteins. It does not release the unwanted proteins. It releases the proteins in the only in a particular number, not in a more number. So thank you for watching. Please do like, share and subscribe to my channel. Thank you.